Meiosis. Meiosis is a special type of cell division necessary for sexual reproduction in eukaryotes. The cells produced by meiosis are gametes or spores. In many organisms, including all animals and land plants, gametes are called sperm and egg cells. Prior to meiosis, all chromosomes are duplicated in a process similar to chromosome duplication, also known as mitosis. It is the longest phase of meiosis. During prophase 1, DNA is exchanged between homologous chromosomes in a process called homologous recombination. In metaphase I, the tetrads are again arranged across the center by the movements of the phanerochores with the two centromeres opposite each other. But this time the sister chromatid will not be pulled apart as in mitosis. In anaphase I, the chromatids holding the chromosomes together loosen. The two homologous chromatids of each tetrad are separated into separate poles. Since the chromosomes from each parent can go into either pole, this is another means to increase genetic diversity in this phase. Like in mitosis, the chromosomes are moved into opposite poles and the nuclear envelope reforms and the spindle is broken down. Remember that there are two chromosomes, not one as in mitosis. The homologous chromosome pairs complete their migration to the two poles as a result of the action of the spindle. Now a haploid set of chromosomes is at each pole, with each chromosome still having two chromatids. Chromosomal replication does not occur between meiosis I and meiosis II. Meiosis I proceeds directly to meiosis II without going through interphase. In meiosis, the cell goes directly from telophase 1 to prophase 2 without the interphase. In prophase 2, the nuclear envelope is again dissolved and the spindle is set up again. Prophase 2 is identical to prophase of mitosis except that there is half the amount of chromosomes. Again the chromosomes move into the center and line up. Now there are two chromosomes instead of two tetrads, so that the chromatids will split off this time. The phanerochores move towards the poles, splitting up the sister chromatids. In telophase 2, the chromatids concentrate in the poles and the nuclear envelope is reformed and the spindle again is dissolved. The cells divide for the last time, leaving a total of 4 haploid cells. Cytokinesis 2 is the process in which the cytoplasm of a single eukaryotic cell is divided to form 4 daughter cells.